Good morning, students. Now coming to the last topic of this chapter, bacteria. That is beneficial activities of bacteria. Students, bacterial cells are highly useful to human beings in many ways. One of the importance of this bacteria, this is bacterial cells. They are highly useful in the extraction of valuable metals. So, metal extraction. Okay, some bacteria they are also highly useful in the extraction of valuable metals like uranium is one of the valuable uranium. This is one of the valuable metal. So, by using these bacterial cells, we can reduce the cost of metals up to more than 50% of the cost. Very, very important. So many bacterial cells they are highly useful in the extraction of valuable metals like uranium. By utilization of these bacterial cells, it can reduce the cost of metals more than 50%. This field of use of bacteria that is considered as biomining. This is called biomining. Very, very important. The utilization of bacterial cells in the extraction of valuable metals like uranium and can reduce the cost of these metals more than 50%. This field of bacteria, this is called biomining. This is one of the importance of bacterial cells. At the same time, the DNA components, okay, the DNA components obtained from bacterial cells, the DNA components that obtained from the bacterial cells, they are also used as biosensors. Okay, they are also used as biosensors. But what is the importance of these biosensors? These biosensors they are highly useful in the detection of biologically active pollutants. Biologically active pollutants. Okay, biologically active pollutants. Very, very important. So these type of very, very important. So the DNA components from bacterial cells, different DNA components obtained from the bacterial cells, they are used as biosensors. These biosensors they are useful in the detection of biologically active pollutants in human body. It is used. These biosensors are used as they are used in the detection of biologically active pollutants. At the same time, these DNAs they are also useful, highly useful in medical diagnosis. Okay, medical diagnosis, and also used in the industrial production of food materials. At the same time, this also used in this fermentation. Okay, bacteria are also highly useful in the fermentation activities. So they are highly useful in medical diagnosis and food and fermentation activities, etc. This is the importance of bacteria. At the same time, bacteria are highly useful in a genetic engineering process. Genetic engineering process. This is also called recombinant DNA technology. How these bacterial cells they are useful in recombinant DNA technology? In recombinant DNA technology, the plasmids required for DNA technology, the plasmids required for this DNA technology, they are obtained from the bacterial cells. Okay, bacterial plasmids means the vectors required for this genetic engineering process, they are obtained from Agrobacterium tumefaciens. Okay, Agrobacterium tumefaciens. This is one of the bacteria. T. I plasmids of Agrobacterium tumefaciens. They are widely used as vectors in recombinant DNA technology to deliver the desired genes into a target host organism. So, plasmid vectors are produced by these bacterial cells which are required for the genetic engineering process. At the same time, the host cells which are required in genetic engineering process. Such host cells are also bacterial cells, especially bacterium Escherichia coli. Escherichia coli. So, bacterial cells, they are not only used as host organisms in genetic engineering process, they are also used for vectors for Agrobacterium tumefaciens. So, bacterial cells, they are not only used as host organisms, they also provide vectors required for genetic engineering process. At the same time, some of the bacterial cells, they improve the soil fertility. These type of bacterial cells, they are highly useful in improving the soil fertility and are highly useful in agriculture. Agriculturally useful bacterial cells, they improve the soil fertility. What type of bacterial cells they improve the soil fertility? How these bacteria they can improve the soil fertility? These bacteria, for example, ammonifying bacteria. Okay, ammonifying bacteria. Example, bacillus. Ammonifying bacteria like bacillus. They decompose the organic materials and make them available to the plants again. At the same time, nitrifying bacteria. Okay, nitrifying bacteria. These nitrifying bacteria, one is Nitrosomonas and Nitrobacter. Nitrosomonas and Nitrobacter. They help in these also improve the soil fertility. At the same time, some of the bacterial cells, they also help in nitrogen fixation and improve the soil fertility. These nitrogen fixing bacteria may be symbiotic bacterial cells like rhizobia present in root nodules of legumes. Rhizobia is one of the symbiotic nitrogen fixer. It is present in the root nodules of legumes and it fixes the atmospheric dinitrogen into ammonia. Thus, they can improve the soil fertility. They can improve the nitrogen contents in the soil. That is symbiotic nitrogen fixers. And some of them may be free living nitrogen fixers. Nitrobacter. Okay, Nitrobacter and also Rhodomicrobium and Rhodopseudomonas. And all these bacterial cells, they can improve the soil fertility. These bacterial cells, they improve the soil fertility. At the same time, these type of bacterial cells, they are also highly useful in the extraction of valuable chemical substances. Chemical substances. Okay, different kinds of bacterial cells, they are also used in the extraction, used in the preparation of chemical substances. These bacterial cells, they are highly useful in industry, in the industrial development of chemical substances. For example, the chemical substance butanol, it is extracted from, it can be produced at industrial level by using Clostridium butylicum. Okay, Clostridium butylicum. This is a bacterial cell. At the same time, vinegar or acetic acid. This acetic acid is also called vinegar. This is produced by using Acetobacter aceti. Very, very important. Acetobacter acetate. This is used in the commercial production of acetic acid. Similarly, another chemical substance, lactic acid. Lactic acid, it is produced at industrial level by using lactobacillus delbrueckii. Okay, lactobacillus delbrueckii is the producer, lactic acid producer. At the same time, propionic acid. Okay, propionic acid. So propionic acid can be produced by using the bacterium Propionibacterium propionum. Propionibacterium propionum. This is a propionic acid producer. At the same time, ethanol, alcohol, Zymomonas mobilis. Okay, Zymomonas mobilis. At the same time, Thermoanaerobacter ethanolicus. This is also on the bacteria. Thermoanaerobacter ethanolicus. Okay, Thermoanaerobacter ethanolicus. And all these chemical substances, they can be produced at industrial level by using these bacterial cells. Butanol, Clostridium butylicum, and acetic acid or vinegar, Acetobacter acetate, and lactic acid, Lactobacillus delbrueckii, propionic acid, Propionibacterium propionum, ethanol, Zymomonas mobilis, and from Thermoanaerobacter ethanolicus. This is the importance of bacteria in a commercial production of chemical substances. At the same time, some of the bacterial cells, they also produce valuable antibiotics. Very, very important. These have many applications in a medicinal field. These bacterial cells, they produce the valuable antibiotics. By using these bacterial cells, we can produce these antibiotics at industrial level. Industrial scale production of antibiotics can be obtained by some bacterial cells. For example, streptomycin, antibiotic antibiotic streptomycin. It was extracted from Streptomyces gracilis. Okay, Streptomyces gracilis. This is streptomycin. At the same time, next one is chloramphenicol. So this chloramphenicol, it is obtained from Streptomyces venezuelae. And another valuable antibiotic, this is Kanamycin. Okay, Kanamycin, Streptomyces Kanamyceticus. Okay, Streptomyces Kanamyceticus. At the same time, another valuable amphotericin. This amphotericin antibiotic is obtained from the bacterium Streptomyces nodosus. And all these valuable antibiotics can be produced at industrial level by using this type of bacterial cells. And these antibiotics, they are highly useful in controlling of the diseases in human beings particularly. So, streptomycin from Streptomyces gracilis, chloramphenicol from Streptomycin venezuelae, Kanamycin from Streptomyces kanamyceticus, and amphotericin Streptomyces nodosus. So, bacterial cells, They are highly useful in extraction of metals, biomining, and also highly useful as biosensors in the detection of biologically pollutants. At the same time, highly useful in genetic engineering process as vectors and as host organisms, and also improve the soil fertility by nitrifying bacteria, ammonifying bacteria. These improve the soil fertility by nitrogen fixation. At the same time, these bacterial cells they also produce different kinds of chemical substances like this chemical substance. Matching question very important. And also produce different kinds of antibiotics.